Hello and welcome to Eyes on Research, the podcast that digests the latest scientific innovations in eye research using simple and understandable terms. I am your host, Thales Guimaraes, medical doctor and clinician scientist at Moorfields Eye Hospital. Hello, my friends, and welcome to another episode of your favorite science communication podcast focusing in the eyes and the vision. I hope you're having a great week so far. In this episode, we'll be talking about the evolution of the eyes. And the idea from this episode came a couple of weeks ago. I was scrolling through social media and I saw a short video on two teams playing badminton. I think it was Canada against China, if I recall correctly. And the speed that these badminton players were playing and reacting to felt almost unreal. How did our eyes evolve to become this quite sophisticated and highly specialized optic system that can acquire information such as this in real time, process it, and send to the brain for interpretation? Well, buckle up because we'll take quite the journey now. Let's start by talking about the structure of our own eyes. The human eye is quite complex. We have specific structures within the eye that make it work or act like a camera. It certainly resembles a camera in many ways, and I'll show you how. It all happens by collecting and focusing the beams of light onto the retina. That's the crucial process to see. The retina is a film-like tissue that we have in the back section of our eyes. But for us to see well and clearly with the high-resolution vision that we have, the beams of light first come through the cornea, which is a dome-shaped, transparent structure that acts like a big lens. Then it passes through a pupil aperture, which is very similar again to how it works in pinhole cameras, subsequently passing through a lens that we call the crystalline lens. Now this lens will focus the light rays to the retina, which has many millions of light-sensitive cells. These light-sensitive cells are essentially the functional unit of the eye, the functional unit that allows us to see, containing two types of molecules that are bound, linked to each other in a membrane. The opsins, which are light-sensitive pigment proteins, and a chromophore, which is a pigment that reacts to light, initiating a change of the receptor protein that triggers what we call a signal transduction cascade, generating electrical activity. This electrical activity will be sent to the brain via the optic nerve. In a nutshell, that's how our eyes work. But how did it evolve to become this amazing optic machinery? How did we get to such an intricate design? And I think the first thing to discuss here is that all animals that have eyes share much of the same genetic information, much of the same blueprint for eye development. We are talking about many millions of years of evolution now. And this suggests that the ancestors of eyed animals had some sort of device, some sort of cell that allowed them to sense light, right? And we do have a master control gene in the eye named PAX6. It controls the development of eyes all the way from fruit flies, octopus, mice, men, etc. Yes, we have our own version of the PAX6, which is extremely conserved through evolution. And when we have genes such as this that have been these highly conserved through the evolutionary tree, this usually means that they are crucial for some sort of function in the organism. The earliest predecessors of any structure that resembles an eye were small photoreceptor proteins that could sense light, or they were light sensitive, as we like to say. These proteins are found even in unicellular organisms, and we call these structures eye spots. They don't give any sort of light direction or vision per se, but they allow the distinction between darkness and light. This is already a huge evolutionary advantage. Perhaps it's an animal that's in the water and there's a predator passing above it, and it creates a shadow, it casts a shadow in, in the sea. So the animal will realize that something is passing through, right? So it will, the evolutionary advantage that this creates is that it allows them to survive. These eye spots then evolved to more complex systems in multicellular organisms, as these now multicellular eye patches gradually form the cup, right? Now, what this does is that it grants the ability, though limited, to distinguish light in different directions. Because think about this, the angle of the light coming into the cup and hitting the photoreceptor cells would tell the organism from which direction the light was coming. And this gives limited, but it gives directional distinction. This is another very significant evolutionary advantage, right? As this pit, this cup, composed of multicellular cells deepen even more with time, these organisms were able to gradually have finer and finer direction of where the light was coming from. After this cup formation, or this invagination if you must, in the region where the light sensitive cells were, it came to a point where making the cup deeper would not allow for a better perception of direction, for a better sense of direction. It is a great mechanism, but a quite limited one. Reducing the width of the opening through which the light comes through became much more efficient at increasing the visual resolution, right? Because now the light needs to pass through this hole. This not only allowed for an even finer sense of direction, but also allowed for a very rudimentary shape sensing, right? The organism could now differentiate some shapes. This is the first evidence that we have of a pinhole. It still did not provide any vision with meaningful resolution because it did not have a lens or a cornea, but again, it is a huge evolutionary advantage because the light comes into the eye more precisely, given it now needs to go through this small hole. Now we are talking about a cup that, remember, is still open. So parasites may come in, contaminate, or cause some sort of infestation in these cups. So the next step was the development of some sort of transparent conglomerate of cells that would allow not only for light to pass through it, because it was transparent, right? But would also protect and allow the contents of the cup to be separated in chambers. We call this the vitreous humor. The next evolutionary jump was the development of a lens. You see, in an eye that does not have a lens, light coming from the environment hits the back of the eye, where the photoreceptors are, where the light sensitive cells are, about the same size of the eye aperture, the pinhole, right? So it depends greatly in the size of the pinhole. And a lens would help focus this into a very specific spot in the retina. So you would take light that's coming from every direction and focus on a specific spot in the back section of the eye. And you would think that making a lens is very hard, right? It's a huge skip, a huge evolutionary skip. But actually, making a lens is not that difficulty. It's not that difficult at all. Any set of transparent material will do the job quite nicely. For instance, some organisms use calcite, which is an extremely common mineral. Adding a lens to this equation makes all the incoming light to be refracted, becoming more concentrated on a smaller area, while keeping the same overall intensity of the light stimuli. So now we have an eye that more closely resembles our own. An eye with a pinhole, a lens, an intermediate transparent substance that we call the vitreous humor, with light sensitive cells at the very back of the eye to process the photons of light. This provided reliable resolution, sense of direction, and shape. What happened to some species then is that a transparent and a non-transparent layer, named the cornea and the iris respectively, also formed. This allowed for another separation of these layers, which formed another chamber. Just like the intermediate chamber that contained the vitreous humor, another liquid, this time is a bit more liquid form, named the aqueous humor. And these new structures were a superb addition because it increases the refractive power of the eye as a whole. Just to give you an idea, the refractive power of a normal human cornea is around 42 diopters. Contrast that now with the crystalline lens, which has around 21 diopters of refraction. In fact, the cornea is responsible for around 2/3 of the total eye refractive power. And the iris, which is the colored section of our eyes, allows for blood vessels to come through this tissue, increasing circulation and allowing for an eye that's even larger in size. Now, this eye, functionally speaking, is identical to the eyes of most vertebrates, including us, including humans. But of course, further evolutionary pressures will drive small changes here and there. So, in a nutshell, our eyes came through a long, very, very long journey to become what they are today. It started as a single light-sensitive cell, which was plain and only enough to give the organism a sense of dark and light. Afterwards, a cupped form, an invagination formed, and gradually became deeper and deeper and deeper. This gave a sense of directionality to the light. Then a small pinhole formed, which allowed for a very crude sensing of shape and more precise light direction, which was further refined with the development of a lens. The eyes then evolved to form specific chambers, which include two transparent humors, an iris, and the cornea. And thus, we arrived in the human eye. Now, these changes are very gradual. The way that evolutionary theory works is that it needs to be a slight advantage over the other, no big skips, right? And that's exactly how this went. Every single step in this process allowed the organisms to have a little bit of a higher evolutionary advantage, and then it became something that resembles our human eye. Thank you so much for listening to this quite amazing story of the eye, I should say. I hope you found this episode useful and that you can extract something from it. Find us on @EyesOnResearch on your favorite social media and send us a message. Would be very nice very pleased to hear from you. You'll also find a link in the description of this episode to our support website, which is the website that we use to post our newsletter. Consider subscribing to us there. It is completely free, our newsletter, and you see weekly articles about curiosities and trivia about the eyes and vision, as well as hot research updates on topics that did not go on to become a full podcast episode. So do consider subscribing. For this, it's a little bit hidden. You need to click the three dots in the top right corner of the website, provide your email address, and hit follow. In this page, you also have the option to donate or become a member, which is completely optional, but highly appreciated. And it does give you access to special posts and very cool things, like, for instance, special posts and extra short podcast episodes where I dive deeper into some of the topics that I discuss in the episode, but with a little bit more detail, as well as allowing you to suggest themes for blog posts, short videos, or even full podcast episodes. Otherwise, if you like our work, it would really help if you could give us a rating in whichever platform you are listening to this episode on. Thank you very, very much. I hope you have a wonderful rest of your week, and I'll see you in the next episode. And as ever, keep your eyes on research. Bye bye.